All right. So just as we did with the cell lectures, where we talked a little bit about cell review and just trying to bring some nomenclature back, right? That's probably more familiar to you than some of the genetic terms. You probably had a cell course and things much sooner than you've had a genetics course. But I think anytime that we start a course, it's good to bring some terms back, kind of bring them to the front of the head a little bit. So I want to do that with what we would call modes of inheritance, right? I want to just kind of bring those up. So let me share a screen here and see if I can get it to do it. All right. Where are my share screens? Okay, I want to share that one. Okay, just one little second here. There we go. We're going to share the screen with you. All right. Okay. So I want to review. This will be a couple of I'm gonna break it down so you don't have to listen to it, 50 minutes worth of it at one time. But I want to talk a little bit about these modes of inheritance, right? Modes of inheritance, that's a term we use to explain how we go from the genotype to the phenotype, what the phenotype is going to look like for a given genotype, right? We talk about how that forms. So it's a term that tends to be found primarily in genetics, right? So let's think about some of those major modes of inheritance, right? And try to find some examples if we could. So if you went out and you ask anybody, I mean, 10 or 12, 14-year-old or whatever, that's had some genetics, a little bit of genetics in high school or wherever, and you ask them, give me a mode of inheritance. Well, they probably know what that means, but they might say, how does eye color inherit it? Or give me some genetic basis for any trait that you want. And probably, most likely, they'll tell you that there's this thing called dominant recessive. Okay, we call it dominant recessive or simple Mendelian because Gregor Mendel was the first one to really bring it forward strongly and explain how it worked. So our first mode of inheritance that we would talk about anywhere in genetics is dominant recessive system. A simple system. We're going to talk about, for all of these that we're going to deal with today, we're going to talk about a one locus, so one gene, two allele system, right? So that you get one from paternally derived, one maternally derived, one from mom, one from dad. And it just depends on how those interact. Those pieces of genetic information that determines the mode of inheritance. So in a dominant system, no matter who you got it from, okay, if you get one copy of that particular genetic sequence, it dominates over what you got from the other parent, right? If you get two dominants, you have both. But if you get a recessive over here from mom and you get a dominant from dad, then the dominant will express itself. The only way you can see the recessive phenotype expressed is to have two copies. Both maternal and paternal genes must have the same recessive code for you to see it, okay? And just classic examples that we always put out there. Things like attached earlobes. Attached earlobes, that's whether the bottom is attached or not, is put forth as being dominant over detached earlobes, which are not. Things like widow's peak. Okay, I can't illustrate it, so I picked little Eddie Munster here to show us off. This is, of course, exaggerated, but the little idea that you've got hair that forms in the front, it's called widow's peak, and that's thought to be a recessive. Same thing with whether you have a regular thumb or what's known as a hitchhiker's thumb, which the thumb is bent back more like this, with the regular thumbs being the dominant. These are all classic traits that you often have people do, and there's lots of these, right? There's things like you may have arm folding, whether you form it over the top left or over the right, like that. Bent pinky, whether this pinky is bent up. Now this is an extreme example here, but the basic idea. Dimples, right? Whether you have cheek dimples or not, present forms, those are cleft chin. You can't see, I don't have one. A smooth chin or a cleft chin like this. You have Darwin's turbuncle, which is this little idea that there's some people just have a smooth head on the ear back here. Some people have this little bump that's either on the inside or the outside. This is kind of on the outside. The other people like me, I'm on the inside. That's thought to be a recessive trait. Okay. Tongue rolling, always like that one because when I teach general genetics, I can stick my tongue out at students for all the time that I can get away with it. But ideas can roll my tongue or not. Okay, and the idea is whether that's a dominant or recessive trait. And there's lots of others. But these are all good, right? They're used over and over again to introduce this concept of dominant recessive. There's usually in a classroom or something, there'll be both two phenotypes. A few people can't roll their tongue, a few people can. It'll be a system. Some will have this ear, some will have a chin. If you play around with arm folding, etc., you'll see that people do it different ways. So they have different phenotypes. And these are all thought to be and proposed to be single gene dominant recessive traits, with one side being dominant, the other version being recessive. So we have things like these attached earlobes that we looked at before, okay? Beta, which is idea that if you drink, these are a little more rare than that, but this is if you eat a lot of beets, okay? Beets are red, obviously, then you have a reddish urine, and some people do and some people don't. A couple of complicated ones, obviously, eye color and hair color are often put forward as having sort of a dominant recessive, with brown eye color being dominant over blue, over green, and then hair color, of course, dark hair color being dominant over others, there's dominant recessive there. But it's very confusing. When the web was young, and you weren't probably around at the time, but when the web was young and you had to program your own little websites, I wanted to learn the programming language. I was a programmer for other things anyway, so I thought, I'll do this web thing, I'll figure it out, use HTML. So I programmed a little series of little pages, ask a geneticist, right? And I said on the page, by the way, I'm not primarily a human genetics person. I do know some about it, but you can ask me anything. And it's really for K through 12. And one of the most common questions I would get would be about eye color or something, right? Both of my parents are blue-eyed, and I've got brown eyes or hazel eyes or something. Am I adopted? And I just say, no, it's not yeah, no. If you look around and read the books, it says that blue is recessive to dark-colored eyes. That's true, but there's at least there's multiple genes involved. Same thing with hair color. It's very different. So I was always explaining, no, it's a little more complicated. Anyway, those are always put forward, and you'll see them, if you look in books, they're there. Things like hair whorl, so in the back of your hair, whether it turns to the right or to the left, is one that's there. And, there's a dominant recessive side proposed for those different phenotype expressions. People do weird things with these, like with handedness, whether you're right-handed or left-handed, people tend to look at other things, like whether that's associated with the certain side of the brain or functioning, artistic ability. That right hand, left hand has been looked at as whether there's a preponderance of it for a sexual preference, okay, sexual orientation. Same thing with hair whorls. People went out and gone to the beaches or somewhere where it's a predominantly gay population, and look to see if there was more whorls going one way or the other. People just tend to do that. I don't know why I think it's got any correlation there, but they often do. Hand clasping, whether you clasp with your left thumbs on top or right thumbs on top, just naturally put your hands together, don't look at them and see which way they go. And it turns out that this left side over here is often the one that's on top, predominant. So it's thought to be a dominant over the right side, which, you can do both, but you feel comfortable one way or the other. We already talked about hitchhiker's thumb, things like mid-digit here. This is mid-digit here, and whether there's lots of hair there or not. And toe length, whether, I don't pull my ugly toes out here to show you, but whether you've got your big toe, and then whether this next toe, the second toe, is longer or shorter than the big toe, is one of the models there. And widow speak, things like that. Okay. Often used, very, very good. You can go into class, and I'm not going to embarrass anybody if they fold their arms or if they have dimples or don't have dimples, and you can say, oh, look, there's, you've got this, or whether and kids like to stick their tongues out, so you can do those as things. And that illustrates, there's two types. There's people who can, people who can't, people that do have this and don't. No difference in them. It doesn't mean that they're odd or anything because they can or can't do it one way or the other. It just means that they're different, and it's a good example of that. Okay. Problem with it is that it's all bogus. Okay. I've lied to you, and pretty much everybody's lied to you. There's almost no evidence. In fact, there's counter evidence looking at familial data and twinning studies that there's no simple gene, genetics of any of these traits. Okay? Um, Now, it doesn't mean there's not a genetic basis to them. There is a genetic. You see people that will, in the same family that can tongue roll or something, and others that can't. But it's not a single gene, and it's certainly not a simple dominant recessive. There's very often multiple gene systems involved or whatever. So you've basically been lied to for a long time. And you'll find that out. And the importance of that is, if you're trying to use this, teach it. Yeah, it's a good example of tongue rolling, who can do it and who can't, but it's really not truly a dominant recessive mode of inheritance. So you have to be a little careful about it. So, okay, what are, what are? Well, there's a lot of them that actually are, okay, that have single locus predominance with dominant recessive. Asparagus urine. Anybody eat asparagus? Many of us know that if you eat it and fairly quickly, if you excrete that, if you have, urine will have a smell, and that's a sulfur-based smell very often. It's methylmercaptan. It smells, if you really get the strong part of it, it smells kind of same thing skunks put out, really, though that's not what the asparagus part is there, but it's got a similar smell to it. If you wander around Funchess Hall and some of the other chemistry labs around campus, you may in fact smell it. Oh, this is terrible smell in the hall, because it just takes a tiny amount. And if it gets out, if you open it outside of the hood, it's not going to hurt you, but it really, really stinks. But apparently the excretion of that is done by gene on chromosome 1. And there does seem to be secretors and non-secretors in a fairly, mostly dominant recessive mode. One that is, and we're going to talk about this later, earwax, whether you have waxy or dry earwax, is definitely under a single gene, predominantly single gene control. Also relates to other secretions in the body, sweat, things. And actually has a cool evolutionary history that we'll talk about as far as what parts of the world, what populations do or do not have waxy or dry earwax in a dominant system. And other things like PTC tasting. We know that this is, while there's several genes involved, it's primarily the TAS2R8 gene, which is found down here in the lower end of chromosome 7. And that's a gene about transport gene. Okay, so primarily, so it's one that transports lots of things that end up tasting bitter to us. So it's in your tongue and mouth. And so, if you taste that, so that's the one where you take a little piece of paper and you put PTC there and go, oh, or people don't respond at all. Another one that probably is in that same family, probably because of this same gene region, is things like cilantro. You have coriander, which is the dried version, but cilantro, fresh cilantro, which I love, and many people love it. Good friends of mine, if they taste it, I can't stand it. It tastes like soap, is the way they describe it. And lots of people say that. And we used to take students into Costa Rica and places, and they would have lots of cilantro. I mean, every meal, the salads or something would have cilantro. And you could almost tell, it was almost a 3-to-1 ratio. It was really fun to see people love it, or people like me who are just, cramming it down as fast as you can. So I think it's probably sort of related into the same form. Point is, there are some dominant recessive traits, many of them, and we'll talk about them. And certainly, if you get away from the phenotype, the full expression, you get down to the gene level, there's a lot of dominant recessive type systems that work there at the single gene level. Okay, what about some other things? Just one example I think is useful here is one of them that's talked about is freckles and red hairedness and this melanin production. Red hair and freckles have lots of things. It turns out they're not single gene controlled. Okay. There's multiple genes there, but one of those fine examples of where you have a lot of things going on. We remember the term pleiotropy. We talked about single gene affecting lots of different traits. Well, the melanin is one of those systems. In fact, even with redheads, it affects pain receptors. Those of you going into dentistry or medicine will learn that people that have red hair tend to require more anesthesia than people who don't, right? They tend to not be quite as tolerant of pain, or they are, in a positive way, they're more acute and aware of pain. But the genes that are involved there in the production of these traits are really part of the melanin system. And there's one that's really kind of cool, and I just pulled it out because I like various mammals. Found on chromosome 16, way down at the end down here, is the MEC1R gene. And this one is a gene that's involved in cellular transport, helping things get across the cellular membrane. So we'll pretend this double thing here is a cell membrane. This is a protein that interweaves back and forth across. And part of its job is to help transport things across. And those things that are transported across become signaling proteins to go signal other systems that eventually end up in the nucleus and ultimately will trigger production of other proteins that are brought back into the nucleus and then affect things like melanin production and melanosomes. And variation in that causes variation in different things. One of the regions here, this is a little example, kind of on the tail region here on the inner surface, so it'll be off here somewhere. There's known variation there that affects the color of coat color in pigs and sheep, as well as foxes and other things. If you go further down into the middle here, some of the deeper parts, there's variation there that's known to affect in mice, like cows and pigs and chickens, and I don't know why it says banana coot is a bandicoot, that's what it's supposed to be. And then others like sheep and mice, other versions of it affect things, okay? As well as humans, okay? It's transporter protein. So the point here is that you got, yeah, you can have single base mutations that affect things in one species, but they don't affect the genes directly. In this case, they affect something else that affects something that affects something that ultimately causes the phenotypic expression for it. One of the cool ones, though, is things like jaguars and jaguarundis. These are found in natively from all the way from southern Texas and into South America. The jaguarundis is black cat, okay? And then, of course, jaguars, we all know what those look like. Well, the color morphs there, even though they're different species, the color relationship there is really due to a series of deletions and mutations that occur right in this shaded region right here, with the black being a dominant form, and then this jaguar color, this mixed color being another form. If you mix them together, okay, even though they're different species, they will hybridize. Sometimes you get the black predominantly coming out. So it does perform as a dominant. Okay. All right. Well, those are dominant recessive. Okay, and we'll just let those go into the background for a moment and pick up a couple more of these modes. One is codominance. Okay, codominance is where, okay, now if I get two different pieces of information, one from each parental unit, how do they behave? Well, one doesn't dominate over the other. They actually both express simultaneously, and you get expression of both. Okay. If you got yellow and pink, then whatever your phenotype it produces is going to have some yellow and some pink, okay, and splotches or whatever. And for humans, I think probably the easiest ones to think about are ones that we use all the time are this ABO blood group, and we're talking particularly about A and B alleles here. If you get an A and a B, then you have blood type, the antigen antibody reaction for AB. They both express. Okay? And of course, B there. Now, interestingly, of course, B is dominant over O and A is dominant over O. So we always use this as kind of an illustration of multiple things. One is that you've got multiple alleles, got A, B, and O at least in humans, and variants of those as well. And you get different modes of inheritance, right? Even though they're the same locus, the same gene, depending on the genetic variation that you get there, you could have codominance going on where A and B are equally expressed, or you can have dominance where B dominates over O and A dominates over O. So you get all these different patterns that can result. ABO is kind of fun. We'll talk about it more as an example. People have mapped all these all over the world. Gosh, years ago when I was postdocing, one of our projects was to look at the area around Israel and some of those were now Israel, Palestine, others from the blood groups. And what we were looking at was trying to look at the major migrations across that region of the world. And we were using ABO blood group data because what we had at the time. And they were taking, multivariate fancy statistics and peeling off layers over time to try to figure out which directions, because, it was a trade center, right? For the most part, as part of the salt trade, Greek salt routes, what had moved through there at various times. Another one we did was looked at mutable graves. It's now Hungary. One of them is they had gone in and found these grave sites and found artifacts. And they actually could to blood type a number of people from the long bones. And there were two ideas. One was that the kind of the overlord class was centered in the middle of the cemetery, and then the serfs, the other people, were around the outsides of it. Kind of a crescent moon hypothesis, it was called. And then another group was arguing, no, looking at the things that are there, the artifacts, it looks like they're just kind of mixed. You know, they weren't things. So we tested that using ABO blood group, and came out pretty kind of fun. This is one where people mapped, and in this argument, they said, well, no, supposedly humans primarily invaded North America through the land bridge, from the Siberian land bridge, Bering Straits, and during times when there was low water, and came down. But if you look at, say, like the blood type B for people who are natives here, you don't see a pattern that reflects that. I mean, you should see things that look like they match up to the Siberian region or areas here, and we don't. We see, looks more like patterns that would come from oceanic invasion. And in fact, that's probably, there have been multiple invasions of humans into North and South America. One of the early ones was obviously from Pacific. People came into South America and moved up. Other people did eventually come in, of course, across the land bridge. So you get a mixing. But anyway, so you can use the patterns for that. As we talk about later, some of this, when you send your genotyping data, some of mine suggests that I do have an ancestry in Central America here for the same group. Okay, because I have a little bit of Native American, DNA in my system, so it maps out to here. It's kind of fun. All right. Well, what about other things? One other little example, real quick, are microsatellite markers. Whether this is the old gel type, but it shows up. If you use a computer model or a computer version with the lines. But either way, if you get a single band, then you've got a homozygote. You can have this as a homozygote for one mutation, one length of microsatellite. And this is one for a shorter length of microsatellite, so it dropped off a couple of bases. But these are both homozygous. They're homozygous for different forms. This would be big A, if you want to play that game, or big B, or little a, or whatever. Okay. So these four individuals are all homozygotes. Now the two are homozygous for the same thing, and these two are homozygous for the same. And then the heterozygotes, very easy to determine because they get two bands, right? So this one's a heterozygote because it got something from mom that was short, something from dad that was longer, or vice versa. And so we can see the patterns there. So if you want to go, and ask if somebody, that's how it would work. You've done this in lab, of course. Is this potentially a child of this couple right here? Yeah, I mean, well, you don't know whether it is or not, but what you know is that it's consistent with it. You can't rule it out, right? Now, if this had a band that was way off up here somewhere and neither one of these had it, then no, it had to be another individual involved at some level. So this is a complete codominance, right? It's a nice little simple expression. Works out real well. It's used all the time in forensics labs as markers. Okay. All right, well, what about something else? Well, if we move on further, we can talk about incomplete dominance, okay? Incomplete dominance is where something ends up in the middle. You don't get equal expression of A and B, okay? Unlike, on a calico female cat, where you get orange and black and you get splotches of orange and splotches of black. This is a case where if you put those together, you get something in the middle. A simplest example would be you got a gene that's controlling color. One locus is producing white, or absence of color. The other is producing black pigment. You put them together, instead of getting a blotchy black and white thing, you get something that's gray, something in the middle. That would be incomplete dominance. And many things are incompletely dominant. One of them that's thought to be that is things like hair, whether it's curly, straight, or wavy. Often those are individuals they're heterozygous. Now there's more genes involved, but for our purposes, wavy can be considered primarily heterozygous outcome. Okay, so it's incomplete. It's somewhere in the middle. Now it doesn't have to be perfectly in the middle. Okay, you can have very, very curly hair and be wavy. You can have almost perfectly straight hair, but a little bit of wave to it, and be in here. So that they can fall anywhere in this dynamic in here. Things like Tay-Sachs. Tay-Sachs disease is a single gene, at least the predominant form of it. is a single gene locus. I'm gonna let you, somebody will have it on their chromosomes, so I'm not gonna jump into it too much, um, but you have individuals who don't have it at all. They have the one side homozygous for the non-affected allele. If you have the affected allele and you're heterozygous, you're in the middle somewhere. You have expression of Tay-Sachs, but it varies. And then if you are recessive, or you have two copies, I should say, not recessive, two of the copies of the mutated form, then you have full-blown Tay-Sachs. So it's one that's kind of incompletely dominant. And if you're heterozygote, the thing that makes it incomplete is that you're somewhere in the middle. And there's people some people have almost no, even though they're hets and could express it, they have almost no expression that links them to Tay-Sachs. Other people have almost full-blown expression of the trait. So it can vary in various ways. Right? Okay, well, that's all I wanted to talk about for the first lecture. The second lecture, we'll now move on to some more complicated versions of it, these modes of inheritance. Again, we're still just basically dealing with a single locus version. We'll get a little more complicated as we work through it.